So welcome to your lesson now on proteins. We mentioned in the very first lesson when we talked about carbohydrates that you've got a monomer, that's one single unit, and when you click them in together, you end up with a polymer, a chain, right? So the sugars can do that. They can form these polymers of sugar, so we call them, first it's a disaccharide, and then it's the polysaccharide as they get longer and longer, no problem. And because of the way that the functional groups are around the outside of a glucose, you've got all these different hydroxyl groups that you could come off of, right? Nice and easy. So you can actually branch your chains off for sugars. Proteins aren't built like that. You cannot branch off in any direction. You'll see why shortly. But you can only go off in one, so either going left or going right. So you will only have this unbranched, long unbranched chain. That's an important little piece of information. Sugars can branch. Proteins are unbranched. The amino acid can mean one single amino acid, or sometimes they will say amino acid chain. They can also call a single amino acid a peptide. Or if you have a few amino acids together, you could probably still call it a peptide. It gets a little bit annoying. So then you call them small peptides, large peptides, or individual peptides. So the language is a bit annoying, but the other name for it would be amino acid. For the single one is an amino acid. Once you put amino acids together, you get a peptide chain. And then once the peptide chains are created, like what you see here, this would be considered a peptide chain. Then, depending on what the functional groups are for each of these separate amino acids, you're going to form different structures. And then from that, you're going to get these folded or coiled little sections of these peptide chains. And then from there, you can build yourself a protein. So a protein is actually this huge, massive molecule built on how these things are twisted up. But you'll see that shortly. All proteins have the same basic structure, as all amino acids do. They're all unbranched. We have 20 different amino acids in the human body. So all of your proteins, they're made up of a combination of these 20 amino acids. You are actually only able to make 12 of them, though. Your body has the machines that can make 12 amino acids. The other 8 amino acids that you cannot make are going to be consumed in your diet. That's why we call these 8 essential in your diet. That's a type of test question as well. I'm going to ask you, what does it mean for something to be essential? It means that your body doesn't make it, you require it in your diet, and I'm going to ask you how many. 8 out of 20. Now, depending on how old you are, if you were a baby, even though you can technically make 12, that 12th one, histidine, you don't make enough of as you're growing. You're just growing way too fast for what your body can supply. So in some textbooks or in some teachers will tell you that there's 9 essential, but that's only considering the histidine that you need in extra amounts when you're little. You can actually make 12. So please use my numbers. Don't use exercise science numbers or anything like that. Use the 8 out of 12. That's what'll get you a mark in my course because we're considering the adult. So here is our fully formed amino acid. I'm going to go to the other page just so I can draw it out for you and show you how easy it is to draw it. But we will come back to this version of it in a second. So every amino acid has the exact same structure to it. It always has this nitrogen and then a couple carbons. So we have a nitrogen, a carbon, and a carbon. On the nitrogen end, it's an amino group. An amino is NH2. So that's nice and easy. Then on the right-hand side where that carbon is on the end, it's a carboxyl group. It's double bonded to an O with an OH. The middle carbon always has a hydrogen in one of its spots. Remember, it's going to have 2 more bonding sites. It's a hydrogen in one of its spots, but depending on which amino acid you have, that spot that's pointing down right now can be a whole bunch of different things. So we call this the R group because R doesn't represent there's no element on the periodic table that is R, so we just use R for in general. If you look to the next page, you'll see the types of things that we can put in that R position. So here we have our NCC. In this diagram, I want to point out the fact that they've put that H that belonged over here, they have put that over on this side. So they've got a charged version. I just want you to stick with our version where you see that it's a full amino group, not with the extra H, and that this is a full carboxyl. Just please draw it this way. It's simpler. You don't have to consider charges. Okay? But that R group that's here can change depending on what you've got dangling down. So in some cases, you might have something that's non-polar. Why is that important? Whether it'll head towards water, the way that the heads of the phospholipid did, or whether it'll try to tuck away from water. So really, if you've got something that's non-polar on a protein and you throw the whole protein into water, you can imagine that any of these little R groups are going to move towards the inside of the protein. They don't want to be on the outside where the water is, right? That also means then that anything that is polar will be hydrophilic and seek out water. So that will automatically go towards water. The other ones will automatically hide away from the water. So keep that in mind. You see that we've got all sorts of different things. We've got, a carbon with another hydroxyl on there. We've got the sulfhydryl group in this case. We've got ring structures, all sorts of different things. And these are the 20 amino acids that are in the human body. You can also even have ones that are electrically charged. So that, let's say you have a negative one sitting across from a positive one, you can imagine then that they will turn towards each other. But if I have two negative amino acids across from each other, they'll push away from each other. So by having the right R group in the exact right spot, what we're going to achieve is the ability to fold this molecule without having to tell it how to fold. If you just have the right negatives and positives and the right polar and nonpolar in the correct order, it'll automatically, as soon as you throw it in water, fold up, which is really quite brilliant. It means you don't need a secondary machine to make sure it twists. It just does it on its own. Get them in the right order, and you have a perfectly formed and functional protein. Get them in the wrong order, and your protein is the wrong shape. If a protein is the wrong shape, it doesn't work the same. It could be completely non-functional. It could be just not as good as the one that is the perfect shape, or it could be just actually detrimental and it can cause damage. So this is where the DNA mutations, which are the actual code for the proteins, if you damage the DNA, then you can damage which order of the amino acids you have, and if you damage that, then you can damage the function of the protein. So that's kind of where we're going to go in our molecular genetics unit. So that R group that we have here is going to change based on which amino acid we're talking about. Let's flip back to the other one because it's got the full picture. So again, with sugars, what are the functional groups? So if you have a sugar, you got a hydroxyl on the one interacting with a hydroxyl on the other. And today I gave you the name of that linkage, a glycosidic linkage. So hydroxyl plus hydroxyl on a sugar forms a glycosidic linkage. Then we get a glycerol and we get the fatty acid chains. What's the functional group on the glycerol? So hydroxyl on the glycerol, carboxyl on the fatty acid chain. Okay. Forming what type of linkage? Ester. So now that we're getting to proteins, we're going to have an amino group on the one end and a carboxyl group on the other. Memorize that. So it'll be the carboxyl group of the one and the amino group of the next one that slides in that are going to have the dehydration synthesis. We're going to do the exact same thing. We're going to take out of water. So we'll end up taking off this OH group from the carboxyl and then an H from the next guy that's here. And that those two H's in the O make our water. And we'll end up joining this carbon directly to the next N. And it's going to be a very, very simple one to draw. As far as memorizing it, I'm going to ask you to use the amino acid glycine. And if you look at the next page, glycine is the simplest one. It's this one right here. The R group is just an H. So I'm trying to keep things as simple for you as possible. What we've done in this picture, so we've been asked to produce a dipeptide, so something that has two of these peptides or two amino acids. And we've used leucine and serine. And it just happens to be that leucine's R group is this horrible chunk of carbons and hydrogens here. And then serine is this group right here. So notice again, we've got the NCC. We've got our amino group, the in-between carbon, the carboxyl group with an R group sticking off. Then we have an amino, middle carbon, carboxyl group, R group sticking off. Done. It's pretty simple to draw them separate. When we go to draw them together, it's going to be this OH part of the carboxyl. Please, this is not a hydroxyl, right? Because the whole thing is a carboxyl. So don't say it cuts off the hydroxyl. This is a carboxyl here and an amino group that are interacting. And when I take off the OH and the H, that's my H2O. So that means that this carbon right here is bonded directly to this nitrogen now. So now it's NCC, NCC. Draw your carboxyl, draw your amino, double bond your O, get your H's on top, and then for your test, it'll just be H's on the bottom rather than this extra large R groups. I might ask you, though, for a tripeptide. And then just keep in mind that if I form one water by joining two of these, I'm going to form two waters by joining three of them. So you'd have to just make sure you have the plus water as the result as well. So let's add that in. Say it again for the millionth time. When you're joining two sugars together, it's two different hydroxyls forming a glycosidic linkage. When you're forming fats, a triglyceride, it'll be a glycerol with three fatty acids, the hydroxyl across from a carboxyl, and you're going to form an ester linkage. When you are forming a protein, a dipeptide, tripeptide, however long you want your peptide chain to be, it's going to be a carboxyl group across from an amino group, and you're going to form a peptide bond. There are a couple other terms I need you to know in terms of the types of reactions that we're dealing with. I've already introduced dehydration synthesis, same thing as condensation, and that's removing this water in order to synthesize or join them together to create this larger molecule. But the opposite is also true. If I've removed a water in order to join them, then I can bring another water back in order to break them apart. So a massive part of your digestive system is using water in this way with these tools called enzymes, which are also made of protein actually. And the enzyme takes the molecule that you want to break, it takes a water molecule and it shoves it into exactly the right spot so that you can split this peptide bond using that water. The name of that type of reaction is hydrolysis. The word lysis means to burst. So this is water doing the bursting, doing the breaking up or the busting apart. So dehydration synthesis or condensation, to join it, you remove a water to create the bond. Hydrolysis, you add water to break the bond. The other terms are anabolic. You've probably heard of anabolic steroids before. Somebody using anabolic steroids would be using that fat steroid, those hormones, in order to help them build muscle mass. Anabolic means to build. If it's catabolic, I want you to think either catastrophic, which means to utterly destroyed, or I want you to think cats and curtains, also utterly destroyed. And you can say that catabolic then is to tear down. So anabolic to build, catabolic to tear down. That means that any dehydration synthesis reaction I'm building, so it's also considered anabolic, and any hydrolysis reaction I'm digesting, I'm breaking apart, so I consider that a catabolic reaction. Okay, so keep those words in mind as well. Those are also the types of things that end up on tests for terminology type of stuff. Once we've joined these guys together, this is the most basic level. This is called primary structure. So here we have our little tripeptide that we want to draw, made from 3 glycines. So I know that the R group's just going to be a little simple H, nice and easy. And I only want to show the final product as this question is telling me. I don't need the formation. So here's how I do it. I know that NCC is the backbone. So I do that, NCC. I know that's going to join directly to the next NCC. And there's 3 of them, so I do it again. Okay. I know that on the left-hand side I have a full amino group because I didn't mess with those ones. And I know on the right-hand side I have a full carboxyl group. And I know that the NC second C, what used to be part of our carboxyl, so that's still got a double bonded O in both cases. Everything else is a hydrogen. Hydrogen up and down. There's one here as well, up and down. One here, up and down. So all you're doing from this point out is just making sure that you're counting the right number of bonds. Remember that nitrogen has 3 bonds, or I should say bonding capacity. And then the carbon has 4. Oxygen has 2. Hydrogen has 1. So if you're taking care of making sure that you haven't created too many hydrogens, you should be good to go. Again, most of the time when people are making mistakes on this, they get hydrogen happy and they put too many hydrogens. They like to put a hydrogen down here, but there isn't space for one. Nitrogen can only have 3 bonds, so you can't afford a 4th bond. So just don't get hydrogen happy. Just count your bonds and you should be fine. You'll catch these silly little mistakes before you hand in your quiz and you'll get perfect. When I'm marking this, this is literally how I do it. I say out loud in my brain, if that makes sense. I say it to myself, NCC, 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 carboxyl, amino, double bond, H is all around. That's what I do. That's how I mark it. That's how I go fast to go through your drawings. So if you're doing the same thing and you're making sure that those things are there, you're good. You're going to get perfect. Let's move on and we'll talk more about the actual final structure because we haven't created a protein yet. We've just created a small peptide. So let's get to doing the actual protein itself. So now we're going to chat about the different levels of structure that we have in these huge protein molecules. So the first thing that you need to understand is that the function of that protein is based entirely on its shape. If you change the shape, you're going to change the function. Then you need to know that the shape is based on the order of amino acids because those little dangly R groups matter. Which one is where? Do you have a negative across from a negative? They'll repel. Do you have a polar one there? Okay, it'll seek out water. Non-polar, it'll hide to the inside of the molecule. So just putting them in the right order, you're going to get a certain bending or shape to it. So the primary structure is done by the ribosomes. You have ribosomes sticking them together. So the ribosomes are responsible for making those peptide bonds, for taking the amino on the one side and the carboxyl on the other and removing the water. That's the job of a ribosome. So the primary structure then is the sequence of amino acids. The secondary structure, the next level of organization, is probably done in either the endoplasmic reticulum or it could be done in the cytoplasm as well. It just depends which protein it is. So you would have the order of amino acids, and then based on their R group dangling down, you'll either form these alpha helix coils. Please, when you are describing them, call them alpha helix. Don't just say helix, don't just say alpha coil. Say alpha helix coil. What you can do, though, is use the Greek symbol alpha in place of the word alpha if you prefer. Or, depending on what's dangling on there, you might also get these pleated sheets. So beta pleated sheet. And they kind of just go back and forth. And again, that's because of the R group that's dangling off. So primary structure, put together by the ribosomes, just get the amino acids joined so we get our peptides. Then the peptides begin to either coil up or they begin to fold. So they're going to fold up based on what's dangling down or pleat up, depending on. Then you get to tertiary structure. That's a fancy way of saying third level. So the third level of structure that we have is how you fold the actual pleated sheets and alpha helix coils. And this again matters on what little dangly bit is sticking out. If it's polar, it'll go to the outside of the molecule where water is. If it's non-polar, it'll want to tuck itself in. If it's negative versus positive, it'll be attracted to something that's oppositely charged to it, or even something that's neutral, I guess, technically. So you're going to get this folding of the subunit, tertiary structure. And then, once you have a subunit, notice how there's 4 different colors in this particular diagram. They're trying to show this actually is supposed to represent a hemoglobin molecule, which has these 4 little subunits. There could also be other things that get added in. In hemoglobin specifically, you have little iron ions, Fe, 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 and that's going to help you to carry oxygen. So it might not just be pure protein amino acids. You could have other things going on. Remember we saw that vitamin B12 that was chelating or surrounding the cobalt ion. So same thing here. We can add other things in to make this fully functional. So once all the subunits are in place and you have all the other modifications done, which is usually done by the Golgi body, it's going to organize your final structure. Then now you've finally got, after quaternary structure, now you've got a protein. Before you've just had a peptide. Please make sure you know how to spell quaternary. It's not quarter, it's quater. There's no R here. So then if we're looking at it in order, then we say what's primary, secondary, tertiary, quaternary. We can say the amino acid level is going to be just put together by the ribosomes. That's my primary. Then I get my small chains. Then I get these larger chains put together, and then I finally get a protein. And in grade 11, you did the reverse of that. You talked about how proteins get broken down by hydrochloric acid and pepsin in the stomach. And then when they go into the duodenum, then you learned that the pancreas and the small intestine squirt out some juices. Small intestine produces the peptidases that chop the large ones into small chains. And then the pancreas produces the trypsin and the chymotrypsin that end up chopping the small peptide chains into individual amino acids. Now they're ready to be sucked into the bloodstream and taken to whatever cells need to use them. Finally, for the protein conversation, we have this topic of denaturing. You're going to see this throughout our entire course, so learn it now. So you're awesome at it for this quiz and test, and you're good for the rest of the time. Denaturing is the idea that a protein the actual shape of it can be damaged. And it can be damaged either when you change the temperature or when you change the acidity or the pH level. So when the pH level drops and I'm getting more acidic, I can actually begin to damage the shape. And the reason for that is that both temperature and acid can get in and disrupt the side chain interactions that are happening here. So if an acid gets in there and it pushes two amino acids apart from each other, then you're going to have a different shape to your protein. Different shape, you can unravel that sucker. If you're unraveling it, then you are going to have a different function or just a complete loss of function, because how it's shaped matters. Where the functional groups are matters. So if it's denaturing, the protein has been unraveled. Now, specifically when the hydrogen bonds are broken, it could also be the sulfhydryl bonds as well, because the sulfurs across from each other can cause sulfide linkages. But when the hydrogen bonds that are kind of loosely attracting and holding them together, when they are broken, because of a change in temperature or pH, means that it can no longer function normally, but it can return to its original shape. If you take away the change in temperature and you put it back to normal, or you get the acidity level back right, as long as you haven't actually snapped the peptide bonds, and again, the peptide bonds would be these ones here, as long as you haven't actually physically, or I should say, as long as you haven't chemically separated those separate amino acids and broken this bond, then as long as these bonds are all intact, as soon as the temperature and pH goes back to normal, it just refolds like it did originally. And so it can go back to working again. Let me give you an example from grade 11 that you may have learned. In grade 11, you learned that in the stomach, there is an enzyme that helps to digest the proteins called pepsin. Pepsin only works in an acidic environment. When it's created, it's bound up too tightly. It's called pepsinogen because it will become pepsin when you mess with it. So if you just have it in a neutral solution or even in a basic solution, pepsin doesn't work. You have to have it's too tightly coiled and it can't wrap itself around the parts that it has to cut for the protein. But if you can loosen it ever so slightly, you actually click it on. When you throw acid at it, it loosens to the right shape, and now pepsin works. So you have to have the hydrochloric acid in the stomach, not just to loosen up the proteins, but also to loosen up the machine, the enzyme that's going to do the cutting. And when you do that, pepsin turns on. Okay? So it's important then that with denaturing, when you're doing this answer, I'm going to ask you this quite a few times. You're going to give me these exact points. You're going to say, just regurgitate this to me and get your full marks. You're going to say that denaturing occurs when a protein is unraveled. You can also say changes shape. When the hydrogen bonds are broken because of a change in temperature or pH, means it will no longer function. You must have that in there. And then it'll return to original shape as long as the peptide bonds are not broken. So make sure you have all those 4 little details in there, and you'll get your perfect marks. Okay, again, quiz question and test question, and you'll see it on and on and on and on throughout our entire course. Final little example here. You've all had fevers, I'm sure. Every single one of you has probably had a fever. Anyone had a fever that's like significant, 104, 105? If you have a fever of about 100, 101, your normal body temperature in terms of Fahrenheit is about 98.6. That's the average temperature. Some people will run a little bit cooler. Some people will naturally run a little bit hotter than that, but 98.6 is about average. Okay. You will also have a different temperature based on whether you do in the mouth or underneath your armpit or other locations where you can take temperature. There'll be a slightly different number that will be associated with that. If it is in terms of Celsius, it's 37.1 body temperature, so about 37. If you have a prolonged temperature, so somewhere 38, 39 in Celsius or over 100, then over time, what's happening if you prolong that fever, it's a longer period of time, is you actually begin to damage the body's proteins. But a fever is a natural response. It's actually part of your immune response to try to fight off any type of bacteria or virus that's in there, because the hope is that you destroy the invader's proteins and damage them before you damage your own proteins. But it is really important that if you have a fever, you want to bring that fever down before that, if it's prolonged, before that fever damages you. So that's why you'll often have the cold compress on the head or on the back of the neck just to cool your head down. You really don't want to mess with your brain proteins because of the denaturing nature of a fever. We also do this on purpose. If you've ever had a perm, or if you can get your mom or your aunt to admit that they've had a perm back in the 80s, then these hair perms are actually using chemicals in order to, because your hair is made of a protein. There's no cells in your hair. The only cells in here are the ones that are creating the hair follicles right up at your scalp, right? There's no cells here. It's just protein. So you got this protein here, but what you can do is you can use a chemical in the hair perming, whatever they use, and they can create their own bridges between the proteins so that you have this curl. Now it'll relax over time as those bridges that you've created are broken, but in the meantime, using a chemical to denature the hair so that you can get it to sit in just the right spot. That's a little bit more of a permanent solution or semi-permanent solution. People do this all the time as well, though, with a hair curler. You're denaturing the hair to try to get the hair to stay in that curl. That won't stay as long with the temperature change, because as soon as the temperature goes back to normal, those things can go back to normal as well, because you haven't broken the peptide bonds. That's why the curls will eventually relax. Does that make sense? But if you over perm your hair, or you over apply heat to your hair, you can break the peptide bonds, and that's when people's hair gets brittle and it snaps. It's because they haven't just messed with the hydrogen bonds, they've messed with the peptide bonds themselves. That is the end of the formal lesson for the proteins.